Okay, welcome to the fourth screen capture video on the blood. What we're going to do in this video is we're going to talk about the final formed elements in the blood. So we talked about the red cells, we talked about white cells, so we talked about erythrocytes, we talked about leukocytes, and now we're going to talk about thrombocytes. So remember that thrombocytes and platelets, they mean the same thing, okay? These are going to be the smallest of our formed elements. Okay, they're actually not true cells in the sense of their structure. Okay, and that is because these cells they are anucleate. So just like the mature red blood cell was anucleate or did not have a nucleus, so too are these formed elements. They're actually cell fragments from a larger structure. So they are cell fragments from a gigantic cell called a megakaryocyte. Okay, so the megakaryocyte is part of the myeloid cell line. Okay, and the myeloid cell line gives us our red cells and many of our different white blood cells. Okay, but what we can see is in this myeloid cell line we have a gigantic cell that never actually makes its way out of the bone marrow and into the bloodstream whole. What happens is that megakaryocyte, pieces of that cell break off and enter the bloodstream, and those cell fragments are our thrombocytes. Okay, these thrombocytes are going to be very important in helping to clot our blood. All right, so when we cut our cells, it's important that we don't lose large volumes of blood. That would decrease our blood pressure. And it would cause us to essentially go into organ failure. So we need to stop the flow of blood if in fact that does happen. And we're going to use platelets among some other things that we'll talk about to do that. So platelets, just like our granulocytes, are going to contain cytoplasmic granules. Okay, and in these granules are going to be what are known as procoagulates. So when we clot our blood, we try to coagulate the blood or make it thick and sticky. And that way that helps slow or stop the flow of blood. There are some important chemicals that we find inside these granules that are going to help this process occur, okay, namely ADP, so adenosine diphosphate. Okay, you may recognize that as the molecule that ATP becomes after it transfers a phosphate group to a molecule, so that molecule can do some work for us. Serotonin, okay, so serotonin we see is an important neurotransmitter, and also calcium. So these granules are going to contain these chemicals. The chemicals will be secreted, and that will allow the clotting process to take place. Okay, as these platelets float around through our bloodstream, it's very important that we kind of prevent them from sticking to one another and forming a clot. Okay, and we do that by secreting a chemical or molecule called nitric oxide. So nitric oxide is secreted by the endothelial cells, or the cells that line our blood vessels, and what that does is it helps keep the platelets from sticking to the inside walls of the blood vessels. Okay, but we're going to talk about some circumstances when some of these platelets might kind of be able to start sticking to those vessel walls and cause problems for us. If we look at a blood smear here, we can see these little tiny cell fragments here. These are our platelets, okay, and you can see their relative size compared to the size of a red blood cell. Okay, they're pretty small, right? All right, let's go ahead and move forward and talk about this process of hemostasis. So the term hemostasis means to slow or stop the flow of blood. So you see heme in there, that refers to blood, and you see stasis, so that means to slow or stop. Okay, there's going to be three important processes that we're going to talk about. The first is called vascular spasm, and as the name kind of tells you, the blood vessel is essentially going to go into spasm. So if a vessel gets torn, what we can do is, if we have blood flowing through a blood vessel in a particular direction, if we vasoconstrict that blood vessel and we squeeze the walls of the vessel, what that's going to do is it's going to inhibit the flow of blood through that vessel, and it will help to prevent blood loss. Okay, so that's kind of the first thing that we're going to do when we tear a vessel. This is very effective for about 20 or 30 minutes. That gives us enough time for the next two steps to occur. The next step is called platelet plug formation. So this is where those platelets are going to come into play. All right, and essentially what we're going to do is we're going to take those platelets and we're going to form a plug with the platelets in the area where the vessel was torn. All right, and if we take a look at this picture here, we can see that we've got some tissue damage. So right in this area, we can see that the vessel was torn. All right, and when that vessel tears, these underlying collagen fibers, and you can see them in kind of these blue strands, these underlying collagen fibers that make up the connective tissue part of the blood vessel, they will be exposed to the platelets that are flowing through the blood vessel. And the platelets are attracted to that collagen, okay? And they will stick to the collagen, and they go through, so they're attracted to the collagen and they're going to stick to the collagen. And that process by which they stick to the collagen is called aggregation. So the platelets will aggregate, or they'll kind of collect at this site where the collagen was exposed during the tearing of the vessel, and those platelets will then start to secrete those chemicals from their granules. So they'll start to secrete the serotonin, the ADP, and the calcium. Well, what does that do? It does a couple of things. One thing that it does is it encourages the platelets to increase in size, so they get kind of they blow up, I guess, for lack of a better term. They blow up and they become sticky. Okay, and those chemicals, in addition to causing the thrombocytes to increase in size and become sticky, they also are going to encourage other platelets to come to the area and to help stick to that underlying collagen and form a little plug. So we're going to secrete those chemicals. The chemicals are going to amplify the effect because they are going to cause additional platelets that are floating around in the blood to come to the site. Okay, and this is a positive feedback mechanism. The more platelets that we get to the site, the more chemicals we get released. The more chemicals we get released, the more the process amplifies itself. So this is another example of a positive feedback mechanism. All right, let's talk now about the third process in the most complex, excuse me, the third step in the most complex of the three in this process of hemostasis, and that is coagulation, or the actual formation of the clot. Okay, this process, in order for it to take place, we are going to use 13 clotting factors. Okay, many of these clotting factors are plasma proteins, float around inactively in the plasma. One of those clotting factors is calcium, so we're going to use calcium as well. And in order for us to successfully go through this step, all of those 13 clotting factors, they have to be present. If even one of those clotting factors is not there, we're not going to be able to properly form a clot and stop the flow of blood. We can only use the platelet plug for so long. It's only so good at stopping the flow of blood. What we need to do is we need to really kind of stop that flow of blood with these clotting factors. So as I mentioned, many of them are plasma proteins, and many of those plasma proteins are made in the liver. Okay, so you can imagine that it is very important for this process to take place. It's very important to have a healthy liver and healthy liver function. Okay, and in specific conditions, for example, if we have hepatitis, which is infection or inflammation of the liver, if the liver is damaged through drug toxicity or alcoholism, all of this can affect the ability of these clotting factors, many of these clotting factors, to be produced. 4 of the clotting factors are going to actually require a vitamin for their synthesis, vitamin K. So even though vitamin K isn't directly responsible for this process, and you can see this process of clot formation right here. I'll draw a couple of arrows for you. It's a very complex process, and many of these clotting factors, 4 of them actually, require vitamin K for their synthesis. So vitamin K acts in the liver to help these clotting factors be produced. Okay, so the clotting factors, as I mentioned, there are 13 of them. They are labeled, or they are numbered 1 through 13 in the order of their discovery. We can see a couple of them here, right? So here's clotting factor 11. And if we go from one clotting factor, which is 11, and then we go 11A, what that means is that this clotting factor is now active, okay? And when we activate a clotting factor, what we do is we use it as an enzyme to catalyze the reaction and the formation of another active clotting factor, so that we can see that an active clotting factor 11, so 11A, is going to help catalyze or activate clotting factor 9 into its active form. Okay, so you should just be aware that there are 13 clotting factors. We need to activate them in order for them to actually be functional. Okay, and we can see here also that calcium is very important, right? So we can see calcium is used as one of these 13 clotting factors as well. So having a diet rich in calcium and a diet or a functional liver is important to this process. All right. So what we can see is that this process is an extremely kind of complex process. Okay. What we want to do is we are going to do our best to allow these clotting factors to come together in specific ways, and what we're eventually going to do is we are going to convert something called prothrombin, which is PT. All right, this is prothrombin. We're going to convert prothrombin into something called thrombin. Okay, so it's basically taking the inactive form of thrombin, which is a plasma protein, and we're going to activate it. Okay, getting to thrombin from prothrombin is the longest step. So going from the top all the way down to thrombin, this is going to be the longest step of the process. Okay, it's actually not that long. It might take 45 seconds for this process to occur. But it's the longest step of the process. Okay, so what they can do clinically is you can measure your prothrombin time. So that is the time that it takes essentially for all of those, or many of those clotting factors to come together to activate prothrombin into thrombin. Once we do that, once we activate prothrombin into thrombin, we're going to use thrombin to activate fibrinogen. So remember that fibrinogen, we talked about it as one of the important plasma proteins that flows through our blood. It's made in the liver, and fibrinogen is important in this clotting process. What we're going to do is we're going to activate fibrinogen, and we're going to cause fibrinogen, which is a soluble molecule, to form something called fibrin. Okay, and fibrin is an insoluble protein that will start to polymerize. So what that means is if we build one fibrin molecule and another fibrin molecule, they'll attach to each other, and what they'll form is they'll form a large fibrin polymer. Okay, and what we see over here in this picture, it looks like a bunch of red blood cells all caught kind of in a net. What we're looking at here is we're looking at the fibrin, a fibrin mesh network. Okay, and what the fibrin does is it essentially, let me go back. What the fibrin does is it essentially connects our thrombocytes all to each other with an insoluble protein that doesn't allow for leakage of red blood cells out of the damaged vessel. Okay, so the final step, as I have written here, is to convert fibrinogen into fibrin, and then we're going to be able to form this mesh network. Okay, once we form this mesh network, we have successfully kind of formed a clot. Okay, and in the area as well, a lot of these chemicals that are being released and a lot of these clotting factors that are being produced are causing the blood to become thick and sticky, and what that is this coagulation. So the blood forms, goes from kind of a liquid type of a substance, and it starts to be kind of become like gel-like. Okay, all right, let's go ahead and move forward, and let's talk a little bit about disorders of clotting. All right, so it's important that we keep these thrombocytes and these clotting factors moving through the blood vessels without sticking to one another, unless we actually tear a vessel. Then it becomes important that they do stick to each other. However, there are a couple of different scenarios that I want to talk to you about, and these are scenarios when we can form blood clots irrespective of whether a blood vessel gets torn or not. Okay, and that is, the first one is vascular damage. So if we don't actually tear a blood vessel, but we just we cause stress to the blood vessel, we can cause the cells that line the blood vessel to become damaged. Okay, so for example, if I had a blood vessel here, and we need to be aware that this blood vessel here is lined with cells, okay? And these cells are collectively called the endothelium, all right? And these cells are susceptible to the chemicals that are in the blood that are flowing through the blood. They're also susceptible to blood pressure, right? So if we have toxins in the blood, or we have carcinogens in the blood, the things that might flow through our blood from smoking cigarettes, or from eating foods, or taking pharmaceuticals, if some of those compounds are toxic, they can damage the endothelium. Okay, same thing with very high blood pressures. That can damage the endothelium. And if we end up with endothelial damage, but we don't actually tear a blood vessel, we can also cause clots to occur. Okay, and in this case, we have vascular damage, and some of those tissue factors, okay, which tissue factors are kind of little signals that the endothelium, little molecules that the endothelial cells will produce. And what those tissue factors do is they cause platelets to stick to them, okay? And if we have platelets sticking to these tissue factors, so I'll just kind of put a little TF right here, okay? So let's say that the endothelial cells here, right in this area where I have these TFs, is damaged for some reason, okay? What can happen is platelets, which are flowing through the blood, can then start to stick to that area. I'll just write plate, okay? Stick to that area and start to form a clot, okay? And what that can do is it can significantly reduce the amount of space through which blood can flow through the vessel, all right? And if that clot gets large enough, it can totally cut off blood flow through the vessel, okay? So that's one way that we can have clots form when they otherwise kind of shouldn't be, right? And you can see in this picture here, right, we see the vessel walls here have some platelets, some fatty materials stuck to them, and that can encourage, if we have damaged endothelium, that can also encourage fats to stick to our vessel walls as well. Okay, another thing that another hemostasis disorder that we want to talk about is blood stasis. So it's important that we keep our blood flowing well through our blood vessels, because if we allow that blood to sit in blood vessels and it doesn't kind of flow well, some of these clotting factors and platelets can start to go through these clotting steps, okay? And platelets can aggregate. So if we have insufficient blood flow through a vessel, platelets can start sticking together, okay, which is not good. This is one of the reasons why, if you take a flight and you're on the plane for an extended period of time, they always encourage you to get up and walk around a little bit. That helps circulate blood, especially through the lower extremities. The lower extremities, the legs, the calves, are very susceptible to clotting disorders of blood stasis, because blood likes to sit and pool in the lower extremities because it's the furthest away from the heart. It's got to work the hardest against gravity to get back to the heart, okay? If in fact this does occur, if we do start to form some clots in in damaged vessels, or because blood has been sitting in blood vessels and it's not flowing very well, what we form is we can form something called a thrombus. Okay, so what a thrombus is essentially a collection of material that starts to stick on the endothelial wall of a vessel. So here we can see thrombus formation. Okay, if fats start to accumulate in that thrombus, we can call it a fatty streak or a plaque. Okay, this is kind of one of the things that can cause heart attack. So for example, if this is a blood vessel that's supplying blood to the heart, if it gets totally occluded or blocked, that can significantly cut off the flow of blood to the heart muscle, and the heart muscle can die, and that's a myocardial infarction. Okay, well, what if we have a thrombus forming, and then for some reason a piece of that thrombus breaks off? Okay, that thrombus will then become a moving thrombus, which we call an embolism. All right, so here is a picture of the brain, right, and blood vessels carrying blood to the brain. And if a piece of that thrombus breaks off, we call this an embolism. And that thrombus, which is now an embolism, starts moving towards the brain. Okay? And if it's moving towards the brain in this example, it's going to continue to move through vessels that are large enough to carry it. However, what if it happens to get to a vessel through which it cannot pass? It will get stuck in the vessel, and we call that effectively an embolism. So if in this picture it's showing the damaged area right here, if it cannot get through, the embolism cannot get through the vessel at this point, it gets stuck in that vessel, and then all of the underlying tissue in this area will all die, okay, because of the lack of blood flow. This can occur too, so commonly it can happen to the brain. So embolisms can get stuck in cerebral vessels, cause the brain tissue to die, and that's a stroke. It's also very common in the lungs, okay, and this particular type of embolism is called a pulmonary embolism. All right, so we can see up here, this is pulmonary embolism. So the most common types of emboli or embolisms are pulmonary embolisms. So, and the embolism usually starts from a thrombus that is in a lower extremity vessel. So here in the lower extremities, there is a vessel, and we get a thrombus forming. Okay, so we see a clot here. That thrombus then breaks free and forms an embolus. The moving clot then starts to make its way back to the heart, and our veins in our lower extremities carry blood back to the heart. Okay, we can see that here. And eventually that blood goes into the right side of the heart, into the right ventricle, and blood from the right ventricle then gets pumped to the lungs. So we're still going in larger vessels here. Okay, so if I have an embolus in my lower extremity, that embolus breaks off and it moves through larger vessels. It continues to go through larger vessels all the way back to the heart. The heart has large chambers. It enters the large chambers, and then it gets pumped to the lungs. From the lungs, we can see that the vessels will get progressively smaller and smaller as we're delivering that blood to the air sacs in the lungs. And eventually that embolus is going to get stuck in a vessel through which it cannot pass, and we end up, in this case, with an embolism. Okay. All right. So let's move forward and let's talk about how we can deal with blood clots. So as I mentioned, there are natural chemicals that are secreted from the endothelial cells in your blood vessels. These are anticoagulants. Okay, nitric oxide, I mentioned, is a chemical that gets secreted, also a chemical called heparin. They're going to both work to prevent clots from abnormally forming. Okay, we can also give clinically we can give some medicines, some medications that will help to break up clots. Okay, and Coumadin is a very common clot busting medication that will help to break up clots if in fact they do form. Okay, they are going to block clotting factors, okay, by blocking synthesis of them by interfering with vitamin K activity. So remember that 4 of the clotting factors that are produced in the liver need vitamin K to actively work. Okay, so we can see here in this picture down below that warfarin, so warfarin and Coumadin are the same thing. Okay, in order for us to actively produce the functional form of these 4 clotting factors, we need to oxidize vitamin K. So reduced vitamin K will become oxidized, okay, and that helps us form the active form of these 4 clotting factors. Well, what warfarin does is it prevents oxidized vitamin K to become prevents it from become deoxidized and reverting back to its reduced form. Okay, and if we block that activity, we're not going to be able to take the inactive clotting factors and we're not going to allow them to become functional. Okay, aspirin can also be used to kind of disencourage clot formation. Aspirin is going to act as an antiplatelet, so it's not going to work in the liver like Coumadin would do. What it's going to do is it's going to actively inhibit the ability of thrombocytes to produce particular chemicals. Okay, and some of those chemicals are needed in their ability to form a platelet plug. Okay, so this picture over here is just showing you a couple of different types of anticoagulants and how they work. Okay, so they're used to help decrease clotting, right? So anti means against, so we're kind of resisting clot formation. So we can use anticoagulants in many steps of the clotting process, okay, at different areas. We can use aspirin to prevent platelets from sticking together. We can use Coumadin that inhibits vitamin K activity in the liver. We can also use some of these anticoagulants to stop, for example, calcium and other factors, stop those factors from activating thrombin. Remember, thrombin, we need to activate thrombin from prothrombin. Okay, we can also use anticoagulants to inhibit the activity of thrombin into becoming prothrombin and forming the fibrous mesh network. Okay, so we can't get the fibrous net, we're not going to be able to clot the blood. All right, let's take a quick look here at a couple of bleeding disorders quickly. The first is liver disease. So I already alluded to this, and it just kind of makes sense that if most of these clotting factors are produced in the liver, if you have liver disease, these hepatocytes that produce the clotting factor are not going to be able to do so. So we could end up with bleeding disorders. So, you know, it wouldn't be uncommon for someone who has end-stage liver disease, cirrhosis of the liver, severe hepatitis to actually have a difficult time clotting their blood, and cuts for them may be serious. Okay, hemophilias. So the term hemophilia, this is a bleeding disorder. It is a genetically acquired bleeding disorder. Okay, so what that means is our genes are not actually properly producing the correct clotting factors. We may be missing a clotting factor or two because of we genetically are incapable of making the clotting factor. Okay, the most common types of hemophilia are sex-linked. So the genes are actually found on the sex chromosomes. Okay, and they are recessive disorders. Specifically, these hemophilias are found, the defective gene is found on the X chromosome. All right, And these hemophilia conditions, they are often passed from mother to son. Okay, so over here in this image, I just want to remind you what sex-linked recessive disorders are. So remember, sex-linked disorders means that the defective gene is found on a sex chromosome. We know that dad has 2 chromosomes. His sex chromosomes are X and Y. Mom also has 2 sex chromosomes, and hers are X and X. Okay, so why is it that men or boys get this condition, hemophilia, more often than females do? Well, if we have 2 parents that come together to have a child, and the mother has the defective gene, so it's a sex-linked recessive disorder, remember? So what that means is that in order to actually have the condition, you have to have, if you're a male, the X chromosome has to carry the condition. Okay, it's recessive, meaning that if the defective gene is on the X chromosome, the male will get it. If the female carries the defective gene on an X chromosome, both of those chromosomes, both X's, would have to have the defective gene in order for her to have the condition, because she has 2 X's. So what this is showing here is the X chromosome in this female that's red, that's telling us that that particular X chromosome has the defective gene on it. So what are the possible kind of combinations, children combinations, that these people can have, and what would their hemophilia genes look like? Well, we could have a normal son that has a normal X chromosome and a normal Y chromosome. Remember that the Y chromosome is always going to come from dad with the son, and the X chromosome is always going to come from mom with the son. Okay, and in this case, mom gave her good chromosome, her good X chromosome, which is right here, that doesn't have the defective gene on it. She gave it to her son, and he's fine. Well, what if she gave the defective X chromosome to her daughter? Well, here is her defective X chromosome she got from mom, but she has a good chromosome that doesn't have the gene on it from dad, so she is normal. However, she's a carrier for the hemophilia condition. Okay, son, so the daughter is a carrier for it. She does not have the full-blown disease, again, because she has a good chromosome from dad. Well, if we have a son who has inherited the defective X chromosome from mom, and he also got his Y chromosome from dad, this boy is going to have hemophilia. Okay, why is that? Well, since this is a sex-linked recessive disorder, and the defective gene is found on the X chromosome, if the boy gets the defective X chromosome from his mom, he doesn't have another X chromosome to try to mute that particular genetic malformation. He has a Y chromosome, and the Y chromosome doesn't have any hemophilia or any genes on it for producing these clotting factors. So he would actually have the condition. So what this condition is it's the kind of the inability to clot your blood because you have you're lacking some clotting factors. Okay, they could also have a daughter, right? And this daughter would get a good X chromosome from mom and a good X chromosome from dad, and she would not carry the gene for hemophilia. Okay, so that wraps it up for the thrombocyte and hemostasis part of the blood lecture.